Hello. We believe imaging and analyzing cells in their native states are crucial. So, Tomocube develops and presents Holotomography HDX1, which enables label-free, 3D quantitative imaging of cells, tissues, organoids, and even stem cells, without introducing any exogenous labeling agents. It allows us to visualize subcellular structures of live cells with unprecedented details and for a long-term investigation. Previously, you needed to follow several steps for cellular imaging, such as transfection for fluorescent protein expression or immunostaining. It also requires washing, incubation, and fixation. These procedures are not only time-consuming but also inevitably cause phototoxicity or photobleaching. One crucial issue is that cells are significantly modified from their native states and should be avoided for long-term live cell assays in numerous fields, such as neuroscience and cell therapy. Furthermore, the obtained images are qualitative. Researchers do subjective assessments by observing cellular images represented with imaging contrasts with arbitrary units. To solve these challenges, Tomocube presents unique solutions based on holotomography, HD. HD measures the 3D tomography of live unlabeled cells within a few seconds. The reconstructed tomography has quantitative information refractive index distributions, which can be directly converted into biomolecular concentrations. Thus, one can also retrieve quantitative bioimaging information, such as volumes, concentration, and dry mass of subcellular organelles. These quantitative images can also be analyzed with machine learning approaches for in-depth investigation.
This shows a typical image of live cells obtained using Tomocube HDX1. Live, unlabeled and happy cells. This is a 2D cross-sectional view of the measured 3D tomogram of cells from a HEP3B cancer cell line. You can see distributions of a large number of cells with subcellular organelles. You can clearly see the cell and nucleus membrane and nucleoli. The spatial resolution is down to 130 nanometers, and you can see various structures challenging to observe with conventional microscopic techniques, such as mitochondria and lipid droplets. You can even clearly see heterogeneous structures of nucleoplasm and extravesicle-like objects. HDX1 also provides a feature for measuring 3D deconvolution fluorescence imaging with 5 channels and 3-channel overlay. So if you want, you can also easily perform correlative image analysis by combining both holotomography and fluorescence imaging with the same sample. As seen in the right panel, we have introduced DAPI, MitoTracker, and Lipidi to specifically label nucleus, mitochondria, and lipid droplets. One of the powerful advantages of HDX1 is rapid time-lapse imaging, because it only takes 2.5 seconds to measure one tomogram with a size of 220 micrometer by 160 micrometer by 50 micrometer. You can see the unseen biological phenomena. In the left movie, you can see how cancer cells are moving. You can see various intriguing dynamics, such as movements of mitochondria, lipid droplets, and cellular membranes. You can even observe the formation of chromosomes of cells under mitosis. See the left part of the movie. In the right, you see the cross-sectional views of a patient-driven lung organoid. The size of the organoids is about 150 micrometer. You can see individual cells at the subcellular resolution, like the mucosal layer, multiciliated cells, and even mitotic cells in the organoid. This will allow new research opportunities for drug discovery, cytotoxicity assay, and drug screening for personalized medicine. This is a representative study using Tomocube HDX1. We have measured the growths of primary cultures adipocytes for 42 days. Yes, for 42 days, we have images cells in the same field of view. You can see the growth of individual cells and the formation of lipid droplets. Because lipid droplets have distinctly high refractive index values compared to that of cytoplasm, our software can be used for automatic and quantitative analyses. For example, lipid droplets concentration, volume, and dry mass can be analyzed in terms of numbers with real units.
This is the specification of HDX1. A high-end incubator is included. User-friendly software provides streamlined processes for the measurement of images, the reconstruction of 3D tomograms, quantitative analysis, and the management of projects. You can measure cells and tissues in various carriers, such as imaging dishes or commercial multi-well plates. For further information, please visit www.tomocube.com or contact us via info@tomocube.com. Thank you.